Good evening, everyone. Just a couple of announcements I'd like to make. First, I want to thank the SSAB and Dave Hemmelwright in particular for joining us and participating in our annual community workshop that we held 2 weeks ago. Dave came and presented the most recent recommendation that the board submitted to DOE regarding our fiscal year 2018 budget formulation and our priorities. And again, thank you, Dave, for doing that, and thank you to the board for having a role in our annual workshop. I thought the workshop was very successful. We had about 100 attendees that joined us, and we had a very interesting panel discussion with representatives from the 3 prime contractors here on the Oak Ridge Reservation, talking about the role that the Environmental Management Program plays in enabling the important work that's done at the Oak Ridge National Laboratory, as well as the Y-12 National Security Complex, and the future mission of the East Tennessee Technology Park, which is, of course, to become a private sector industrial park. I also wanted to mention that we are continuing to make great progress with the demolition of Building K27 at the East Tennessee Technology Park. This is the last of the 5 gaseous diffusion plant facilities at that site that is being demolished. And when we complete this project, we will realize what we have been calling Vision 2016, which is the safe and successful demolition of the 5 gaseous diffusion plant facilities. We will be having a large celebration to commemorate this historic event. This will be the first time anywhere in the world a gaseous diffusion plant or facilities will be successfully demolished. The celebration will be the week before Labor Day. We're working on identifying which day of that week, and it will be out at the East Tennessee Technology Park. And of course, all board members will be invited to join us and participate in the celebration. So look for that announcement in the near term. And that's it for me with regard to announcements.
All right, now we're into our public comment period. And Laura, if you will bring that open.
The public comment period is now open. And seeing there are none, as a Citizens Advisory Board, the Department of Energy on environmental cleanup, we want to encourage the community at large to attend and participate in our board and committee meetings. If you are unable to attend, please send your questions and comments to the address shown. Appropriate your comments and questions will be read during the public comment section of the SSAB meeting, and we will be given the same consideration by the board and DOE as those given in person. Meeting schedules and ways to communicate with the board can be obtained by calling 241-4583 or by visiting our website.
Thank you, Laura. All right, tonight our presentation is going to be on DOE's technology development to support mercury cleanup strategy. And our presenter is Mr. Mark Peterson.
It's a pleasure to be here, and a pleasure really to talk about mercury technology development in East Fork Poplar Creek. It's a favorite topic of mine lately, and it's a real pleasure. And I appreciate all the folks who have come to hear about this on a nice day out to come in and listen to a technical talk. That's really great. So thanks for coming. Before I get started, I want to just thank all the folks that are engaged in this program. I say this a lot because it's true. I'm very much a messenger. This is really a team approach from managers to engineers to scientists to technicians, a number of folks who are involved in this program and helping it be successful. So thank you to all those folks. So a little bit of introduction of what I want to talk about. I'll just give you a little bit of a background about what we do at the Aquatics Lab at ORNL. And then get right to the main topic and talk about mercury in East Fork Poplar Creek, and specifically on the technology development program that we've started here recently. And as part of that discussion of the technology development, I really want to take quite a bit of my time to talk about the strategy. A lot of effort went into the approach that we took, and I think it's a good one, and I want to kind of lead folks through that a little bit. But then also get to some of the key findings. We started this project back in August of 2014, so it's been about a year and a half, so we have some results I can share with you tonight. Okay, so I apologize, it's a little hard to see here, but just to highlight that aquatics research, it may be surprising to some of you, goes really back to the very beginning of the, shortly after the Manhattan Project. There were researchers out looking at radioactivity in the nearby streams. Then in, let's see here, get this working. Let me try something else here. And then in the 1950s, there was actually a field research station built on the Clinch River to evaluate various contaminants in the reservoir. Then it was in the 1960s, the Walker Branch watershed started, a very famous watershed, 50 years of research going on. The NEON group, Orenell, been involved there. The Aquatic Ecology Laboratory was built in the 1970s, and I'm glad to hear that there'll be some folks coming out to the lab next week to get a tour of the facility. And then in the 1980s, the Biological Monitoring Abatement Program started. Some of you may be familiar with that program. 30 years of assessment of various streams, including East Fork Poplar Creek, and of course, evaluating the mercury issues there. So the bottom line, a very rich history of aquatics work, and especially focus on the local environment and trying to improve it. So today, the lab is really one of the premier aquatics facilities. It's one of a few in the Southeast. We're again focused on solutions to the U.S.'s most challenging energy and environmental related problems. It's a large facility with a number of people working in it, and we're very closely tied to a number of the various organizations here locally in trying to improve the environment. Okay, so we've got a little bit of background on why we're interested in mercury. Of course, we have a mercury issue here in Oak Ridge, and that really started primarily back in the 1950s when there were large quantities of mercury that were released from Y-12 into East Fork Poplar Creek. It was in the early 90s that there were some major CERCLA actions, so various RIs, feasibility studies, a record decision issued, and that remedy focused on the floodplain, and that's really important to understand. It was not focused on the boundary within the creek. It's not focused on the stream itself, but focused on the floodplain. And as part of that investigation, a lot of mercury was removed from the floodplain in East Fork, around East Fork. And then since that time, relative to the East Fork environment, most of the various actions, remedial actions, abatement actions, have focused on within the Y-12 plant, which of course is at the headwaters of East Fork. And then it was in 2013-14 timeframe that there was a number of workshops, a number of interactions with regulators and other stakeholders where it became apparent that a strategy was needed that not only included the remediation activities at Y-12, but considered the downstream environment, that the downstream environment, that historical contamination was a continuing source, continuing issue in the environment that we needed to look at. And I think another key outcome of that strategic planning was a feeling that technology development, the latest science, was needed to be applied to this very complex problem. And so currently and in the future, the big activities, in my view, for East Fork Poplar Creek and mercury is, of course, the design and construction of the mercury treatment facility. It's addressing the largest discharges of mercury from the Y-12 facility, and also the technology development program, which I'm going to talk more about. Okay, so this is just a general timeline for how these various mercury activities or remedial activities and studies are planned. So the mercury treatment facility, is going to be, are you getting feedback there? Is that my fault? Okay. There's, of course, design and construction over the next few years, and then concurrent with that activity, the Mercury Remediation Technology Development Program. I mentioned it started in August of 14, so we've been at it for a year and a half. But ultimately, we're hoping that these activities will lead to an analysis of alternatives for East Fork Poplar Creek in the next decade or so. So when we were asked to look at how we would develop a mercury technology development strategy, of course it was important to be consistent with the priorities and timeline of the various remedial activities. It makes sense that we're focusing on Y12. It's at the headwaters, and so any kind of cleanup or remediation would really need to follow timeline-wise after those activities or major activities at Y12 are being done. The strategy recognizes that it's a watershed problem. It's not a very discrete source kind of issue, that the watershed is a complex place. There's a lot of different moving parts there. We did an extensive literature review, evaluate a lot of on-site data, and our big goal for technology development, again, is to try to find solutions to remediate potentially lower East Fork that is preserving or enhancing of the natural resources there. Conventional approaches to mercury or to any kind of cleanup action in soils or sediments are very intrusive to remove soils from the environment. And so we want to look for solutions where we maybe don't have to do that but can still reduce risk. So our target ultimately is we want to reduce mercury flux, reduce mercury concentration in water to hit our ambient water quality criteria goals, and we want to try to reduce mercury concentrations in fish and ultimately take the signs down from East Fork Poplar Creek. That's our goal. So we adopted a technology readiness level approach, which is this is an approach defined by DOE for use of technology development across the DOE complex. And the approach really starts very broadly in doing paper reviews. We put out a sort of a white paper, which I've got some copies here for, that kind of highlights all of the various, highlights the approach that we're taking and why we're taking that approach. We're also doing more study site characterization early in the early years, and then over time, increasingly do field and laboratory batch or bucket scale testing, ultimately leading to some sort of pilot, recommended alternatives, and then an action. So this is sort of the overall paradigm that DOE recommends for how we develop technology development. So we're following that in how we're going forward in East Fork. Okay, so we have three major tasks in the Mercury Technology Development Program, and I'm going to take a little time here because I think it's really important. For many contaminants, it's all about source control, limiting contaminant release from storm drains or from a facility site or groundwater contamination. But mercury is very different. And USGS in a recent publication focused on mercury and surface waters across the U.S. focused on three key factors that determine the level of mercury contamination in fish. And this is actually the first sentence in that report, and it's done very well. Three key factors: the amount of inorganic mercury available to an ecosystem. So that is the source, the amount of flux coming in. Another major factor is the conversion of that inorganic mercury to methyl mercury. Inorganic mercury is largely not toxic, and methyl mercury is the more toxic form. So the environment, the surface waters, the sediments, that environment that, the microbial communities in that environment that help convert that inorganic mercury to methyl mercury. And then the last factor is the bioaccumulation within the food chain. And so you really need to have source, mercury methylation processes, and the food chain. All of these interact to force a system either into low levels in fish or high levels in fish. So as an example, out west, there are many sites that have have high mercury loading from gold mining, but then it goes into systems where the water chemistry is such that, and biology is such that you don't have problems in fish. Conversely, there are places where you have relatively low inorganic mercury in environments, say Ontario or places in Maine, where then you have, low pH, you have the dissolved organic matter, you have processes that encourage methylation, and then you have real old fish like lake trout and northern pike. And so in some cases, you can have low source, but you have the water chemistry and the food chain driving a problem in fish. So it's really that combination that helps, that contributes to what we see in fish. And so that's the approach that we've taken in the Mercury TD program, is to focus on, okay, well, what do we need to focus on in the East Fork environment relative to sources and controlling flux? And so that's primarily soil, particularly bank soils and groundwater. And then can we develop ideas for perhaps changing water chemistry to try to limit methylation or enhance demethylation? So we want to look at water chemistry and sediment manipulation. And then lastly, look at ecological manipulation. If we have a different kind of food chain, maybe we can help reduce risk. So that's the major approach that we use throughout in this program. Okay, so one of the things we want to do very early on is understand the system, so we can develop the best technological solutions. And I've used this analogy before. If you're going to clean up a contaminated building, you better understand that building really well before you can decide how you're going to remediate it, how you're going to develop technologies to clean it up. And it's the same way with the stream environment. We don't know what tools we may be able to apply until we really understand what's going on in the system. So initially, we've done a lot of focus on the banks and looking at mercury, soil erosion and mercury releases from those banks. We actually put a kayak into the water from the Outfall 200 at the head of East Fork Poplar Creek and floated it all the way down to the mouth with video cameras on the front and the sides, and examined erosion, foot to foot all the way down the creek. And once we have erosion rates, then we can analyze the mercury in the banks and determine what kind of flux we're seeing throughout the bank. So we looked at evaluating mercury and soil erosion, where we've added some gauging and water chemistry stations throughout the creek to get a better handle on the water chemistry. And then in the biota, we're focusing more on methylmercury. A lot of the work that's been done in East Fork to date has been focused on total mercury. Methylmercury is what we're concerned about from a human health standpoint. So we really wanted to get more information on the methylmercury dynamics. Okay, so let me share with you some of the key results. First off, some of the soil and groundwater source control task. In evaluation of stream banks, I mentioned the kayak surveys. This top figure is showing the amount of flux measured as kilograms per year per 100 meters along East Fork Poplar Creek. So here's Y12, here's the headwaters of East Fork Poplar Creek, and then it moves downstream towards the mouth. And as you can see, the highest concentrations of flux from the banks is in that upper section, extending from about EFK 16 to 22 in that area. Total estimated amount of mercury from the banks is about 38 kilograms per year, which is pretty significant. And as part of these surveys, we discovered that there was a historical release deposit where we really had some higher concentrations than other areas within the banks, and I'll talk more about that. So it's a little bit more detail. This is some work where we looked at mercury concentrations from the top of the bank to the stream at 4 locations. And then within each of those horizontal plugs going into the bank, we measured mercury at various depths within that core. And this is pretty interesting. So if you look at the top of the bank, we're seeing mercury concentrations in the 60 parts per million to maybe as high as 120. You move a little bit of distance further down, and we're getting concentrations about 10-fold higher. And that's related to that historical release deposit. Interestingly, in both of these, for the most part, pretty similar concentrations at the surface of the bank, and then also vertically in, or horizontally into the bank. But then you get down into this core, and we've got concentrations in the 200 to 300 parts per million range right at the surface, and then they drop off and they're not there further into the core. And then at the other site, or the other location in the bank, we've got very little at the surface, and then it increases with. So what this is telling us is you got a lot of variation out there, and it's very dependent on the history of when mercury was released and how it was deposited. But a key thing for technology development is that maybe these levels in the 20 parts per million are not a big focus. Maybe we don't need to clean up all of that bank. Maybe what we need to focus on are these higher concentrations that are making the most difference relative to flux overall in the watershed. So we've been doing a lot of that kind of characterization effort to really focus on where are the primary target areas for potential technology development where you make a difference in decreasing flux. In the water chemistry task, the focus has been on 4 major sort of subtasks. One, trying to prevent methylation, looking at options that might promote demethylation. Remember, microbes methylate, and they can also demethylate. We're also looking at solid aqueous partitioning. If you can keep mercury in the bank or in the soils and sediments and not into the water, that's a good thing. So we're looking at sorbents and other kinds of solutions to try to encourage binding in the environment. And then lastly, we're looking at actions within Y12 where we can maybe look at potential alterations in chemical use at the facility that might help with mercury release. So I mentioned that we've got some water chemistry sampling that is done upstream, middle, and downstream East Fork. And I know it's a little complicated graph, but let me kind of walk through this. These different colors are just showing different seasons. So we've got orange, autumn, blue, winter, green is spring, and red is in the summer. And this is showing rates of total mercury, of methyl mercury. And what's obvious when you look at this seasonal pattern is that if you look at these concentrations, you have higher methyl mercury in the spring and summer. This is what you'd expect. It's when the microbial community is active, warmer temperatures. That's when you're seeing the highest methylation in the creek. And then the solid bars versus the open bars are just highlighting the area in upper East Fork is in solid, and then the open bars are in lower East Fork. And so what's clear is that you get much higher methyl mercury in the lower part of the creek than in the upper part of the creek. And so that line is just showing the ratio there. So around 2 to 4 times higher methyl mercury in the lower part of the creek. And so, why that is under investigation. We don't know why that is that methyl mercury is higher in the creek. One of the things we've been looking at is the sediment characteristics. This is some work that Scott Brooks did looking at using sequential extraction analysis to kind of evaluate the binding properties or of the soil and sediment. And so what this sequential extraction does is that it uses increasingly more aggressive solutions to see how much mercury can come off of the soil or sediment. So an F1, that's just deionized water, anaerobic deionized water. So if you get a lot of mercury coming off in F1, then basically that mercury is highly available to go into the stream and be mobilized. In contrast, F5, you can make the analogy of the tornado kind of thing. You know, F1 is very, very little or very less aggressive, and F5 is the most aggressive chemical treatments to be able to pull. So if it's F5, it's largely bound to the soil and sediment, and it takes aggressive chemical treatment to remove it. And so some key points here. The good news is 97% of the mercury extracted is in those F3 to F5 fractions, which is really good news. That means most of that mercury is going to be very hard to come off. So it's going to be bound to the soils and sediments, in this case, the sediment analysis. The maybe not so good news is that as you move from upstream East Fork to downstream, you see that green line. You get increases in the F3 fraction, which Bloom, who developed this procedure, most closely correlates with methylation potential. So, you know, we're seeing higher methyl mercury in the lower part of the creek, and kind of makes sense that we're seeing the chemistry is sort of suggesting that there is something going on why that methylation is happening. So this is just some things we're trying to investigate to get a better handle on the methyl mercury in the lower part of the creek. And then the last task is the evaluation of the use of ecological manipulation or changes in bioaccumulation. And this graph is just highlighting that, or figure is highlighting that, you know, food chain makes a big difference with mercury. The risk for mercury is not touching the water. It's not drinking the water. The risk is fish. And so food chain drives how much mercury is biomagnified and how it accumulates in the environment. Longer food chains enhance mercury bioaccumulation. And each organism has different bioaccumulation potential depending on what it's eating. And interestingly, the greatest biomagnification step is the step between water and algae. Remember, water concentrations of mercury in parts per trillion levels, and that biggest jump is in the 100,000 to 1 million range jump between what's in water and what's in algae. So if you can control algae, you know, you might be able to change the overall uptake of mercury in the food chain, or perhaps change invertebrates or fish populations, et cetera. So that's what we're investigating is, can we change pathways of exposure? And so just the fish population can make a difference. And you know, we do fish management activities all the time. We manage reservoirs for different kinds of fish. We control fish populations. And in Lower East Fork, we have 3 species of sunfish, and they accumulate very differently. Bluegill largely eat terrestrial insects, and so they have very relatively low levels of mercury. Redbreast, which is at that light green, they're omnivorous, so they eat a combination of invertebrates and fish, and they accumulate a little higher. And then rock bass, which largely eat fish, accumulate almost twice what you see in bluegill. So one of the solutions might be, overstock in bluegill. Create a population of bluegill in the creek, and you've halved the amount of mercury risk in the system. So those are the kinds of things that we're looking at. And similarly, in invertebrates, the percent methylmercury varies significantly by species. And so I've just highlighted 2 species that are common prey for fish, caddisfly and crayfish. They're actually fairly comparable in the total mercury, but caddisfly have very little methylmercury, the toxic form, while crayfish have very high methylmercury. And so, if we can do some tweaks to the environment to be able to emphasize lower methylmercury invertebrates, maybe we can change the ultimate concentrations in the fish. And then, of course, so we've been doing all these various characterization studies, and then we've been increasingly looking at how we can develop the technologies, the mercury control technologies that might make a difference. We've been working with our colleagues in the in the carbon fiber arena at ORNL. We're developing some lignin-based carbon fibers, and we've been looking at some other kind of absorbent mats that might be applied. Those mats could be applied onto the bank and then try to and help prevent loss of mercury from the banks into the water. We've been looking at water chemistry and sediment manipulation ideas, working with the Y12 folks and folks to see if we can maybe tweak some of the chemistry there. And I'll mention too, one of the areas that we've been really kind of excited about is the idea of perhaps introducing native mussels into East Fork. They were there historically. They were exturbated during the years of most impact to East Fork. And mussels make a big difference in water, in water chemistry. I mean, if you look at, this is with no mussels and this is with mussels, you can see that cloudy water, mercury binds to particles. So a lot of that mercury that's in solution that may go into fish concentrations, if it goes into mussels, it could be a good thing. And because one of the things that's also really interesting about mussels is that they accumulate very little methylmercury. They're almost totally inorganic mercury, which means that that's not a viable pathway for risk. So if we can so we're looking at sort of the overall mass balance within the biological community, and if we can push some of that mercury that's in the fish population, maybe into mussel population, might be a good thing. And TWA is very excited about this. They've got a new facility in Nashville where they're trying to restore native mussels into streams across Tennessee. And so I think there's a real possibility there to not only reduce risk, but enhance the natural resources in Oak Ridge. So I think there's a lot of exciting things going on there. One of the proposed things that we're hoping to do is to build a Near Creek research facility, which would be highly desirable to be able to bring East Fork Poplar Creek water into tanks and chambers to be able to do some of these experiments and better evaluate the technologies that we might apply. Examples include sorbents and columns, be able to manipulate water sediment chemistry or make the biological changes. It's hard to do to manipulate the stream itself initially without a large investment. If you can bring water into tanks and do that on a smaller scale, it's a nice bridge between the field and the bench scale type of work that we do in the laboratory and the more pilot scale kinds of evaluations. So again, trying to develop environmentally friendly solutions with that facility. And so I'll just end to say that I'm excited about the work that's going on in Oak Ridge relative to mercury and in general about East Fork Poplar Creek. We've shown through the BMAP program some substantial improvements in water quality in the creek. That's really good news. Some of you may have heard ORNL, it's certainly not in my group. Scott and others were involved in a major breakthrough in identifying the gene that methylates mercury. It's published in Science magazine, and they've been evaluating all of the different kinds of microbes that methylate mercury. So, I mean, these are mysteries for the last 40 years, 50 years that have just been recently identified. That could have application in the future. We've done some work on water chemistry at the Savannah River plant, and some of those activities have worked. We've recently published a paper that where we saw fish concentrations go below the 0.3 target. The ecological manipulation has been applied in Oak Ridge at the P1 pond at K25, and risk has gone down. We've hit our regulatory targets, and we've enhanced the natural resource value. I think most stakeholders feel like that has been a good success story. And then lastly, you know, the city of Oak Ridge with the water storage tanks, that's going to help water quality. It's going to help with the limiting storm flow, which helps with at higher storms, or you get more loss of bank soils. That's going to help things. And of course, the Y12 mercury treatment facility is really good news. So just overall, I'm just feeling very excited and feel like we're really going to be able to have a chance to make a real difference in the environment. So with that, I'll be glad to answer any questions. Thanks for having me.
Okay. We will start with questions from the board. If you have questions, please turn up your tent and remember to use your microphone so we can record things appropriately. All right, Mr. Hatcher, I think you're first.
Thank you for a nice presentation, and some very nice work that's been done at the lab. One thing that kind of popped out on one of your diagrams here, where you're measuring the sampling of the soil profiles above stream water. I don't see how you can use that as a reference frame, because stream level goes up and down day to day many times, or it goes way down in times of drought, it goes way up in times of flooding. And I think that decreases the quantitative nature of the measurements you're making here if you're going to use that kind of reference frame.
Reference stream?
The reference frame that you are using is the top of water surface.
Oh, right.
That's
Okay.
what, this is on page 13.
Yeah, that's under base flow conditions, so you're not gonna get.
Okay, that was not clear.
Yeah, so those are base
Okay.
flow, that's a base flow picture.
Okay.
But Bob, you're hitting on something that's really important to understand, is that a lot of that flux that we're talking about is really storm driven.
Right,
So, much of that mercury that's sitting up high in the bank may not be mobilized except during storm events.
okay, good. I think it's very important to try to
Right.
quantify things as much as you have, and you're certainly attempting to do that.
Yeah, absolutely.
Yeah, thank you.
All right, I think the easiest for me, because I'm not sure who put up what first, okay, Greg Hollis, okay.
Thank you, Mark. One clarification and one question.
Yeah,
On your timeline, you talk about somewhere between approximately 250,000 pounds and 470,000 pounds, and the chart said it went into the creek. That's what was lost on the reservation, correct?
no,
In
that's
the soil-
what's anticipated to gone into the creek.
Assumed.
Yeah,
So
Yeah,
you're
uh,
assuming all that was lost will ultimately end up in the creek?
yeah, that was what was lost from Y12. That's an estimate from what came from Y12 into the creek, that's right.
Okay?
Scott wrote a paper on some of those lost, I don't know if you know,
You guys may be familiar with some of the documents on the historical reconstructions of loss. And at the time that mercury was being discharged from Y12, at the time mercury was being discharged from Y12 during the active mercury use periods, they were sampling the water coming out of Y12. So there was some data to base that on. Best estimate is that about 128,000 kilograms was discharged directly into the creek. That's not total loss. That's directly discharged to the creek. So that's just a fraction of the total loss.
Okay.
Yeah. Also, to help put it in context, we generally say that approximately 700,000 pounds was lost to the environment. And of that, the amounts Mark provided is what is it suspected to have been lost to the creek or the water.
Thank you for the clarification. And second follow-up question, how far down river is this a concern? You know, you talk about Poplar Creek and you talked about the Clinch River, which flows into the Tennessee River. It's all part of Watts Bar Lake, and it goes on to the Ohio River eventually. How far down is this been found?
yeah, we see fish concentrations that exceed the regulatory limit, and it's thought to be related primarily to the Y12 releases. Just a short distance downstream of the Poplar Creek Clinch River influence. And in some years, it's actually below the 0.3, it kind of depends. So we see that the limit of the mercury influence pretty much at where Poplar Creek gets into the Clinch. You know, maybe a little downstream occasionally, but that's kind of where it is. So Poplar Creek is posted, and so there's a mercury concern there as well.
Thank you.
All right, Dennis Wilson.
Yes, thanks for the presentation, it's very interesting. Just a question about the ecological manipulation. If you're taking, for instance, the mussels,
Right.
if you're taking and using them to pull the particulates out, they're gonna bioaccumulate the mercury. They then die, and it goes into the sediments again. Isn't that?
Yeah, you're, in that scenario, you're not removing, and there are folks who are looking at applying mussels in cages, for example, and then essentially harvest them.
Yeah.
You know, and it's removing, and they're very effective. You know, if you look at Melton Hill Lake these days, you know, it's crystal clear because of zebra
Zebra
mussels.
mussel,
Yeah. And so they're very effective at pulling those particles out. You're absolutely right. We're not removing anything, but what we're doing is we're pushing that total mass within the biological community into a place where it's in a different form and a less available route of exposure. And so, again, for whatever reason, it's interesting, the mussels do not accumulate a lot of methyl mercury.
Methyl mercury.
So that inorganic form is just like it's just like inorganic mercury bound to soils and sediments in the environment. That if it's not getting into the water, it's not getting into fish, and you don't have a problem. So it's a matter of sort of pushing and adjusting the sort of the system to minimize methyl mercury and push it and push the total balance to an area that has less risk. And I guess, you know, mussels, at least in Tennessee, they're not eaten by humans,
Right.
Unlike fish. So we think that, and you can run scenarios where you look at a raccoon that might eat a mussel, but again, if that inorganic mercury is high, they're not getting enough of exposure of methyl mercury to be a risk. So you don't have the risk with the wildlife. Humans don't eat them, and yet that mussel is helping change the water chemistry that positive direction. So that's the idea, and that's why it's research. You know, we're looking at that. We'd like to be able to examine that more closely in tanks and examine it, you know, firsthand.
Okay. Second question is on the soil and groundwater source control using adsorbents. I maybe missed something in your explanation. Is the idea to put a membrane on the bank?
It could. Right now, we're primarily looking at the characteristics of the mercury in the soils and sediments and trying to determine, remember I pointed out that a lot of that mercury is very tightly bound.
Yes, exactly.
So if you're going to use a sorbent, it has to have more sorbent capacity than the surrounding soils and sediment if you're going to pull it out of there.
Yes.
So, yeah, there are various, you know, that's sort of getting to the engineering or the pilot scale kind of thing. You could use mats. You could put some sort of barrier there. You could do solutions that perhaps just limit the amount of erosion, whether it's, you know, plant cover or some kind of armoring. But the Zorbent idea right now is primarily focused on laboratory studies, looking at the characteristics. We're bringing water and soils and sediments from East Fork, bringing them in the lab and doing these various tests of various sorbents, both off the shelf and new technologies like that we're now developed, and just seeing how effective they are. So far, the carbon fiber, it seems to be really promising.
Interesting. Okay. Thank you.
All right, Ed Trujillo.
Yeah. Thank you, Ms. Peterson. Great presentation.
Oh, thank you.
I probably understood about 20% of that. Anyway, a question I have is, I think you did mention that the floodplain had been cleaned up before?
Remediated, that's right.
Remediated? Portions of it.
Right.
Okay, so do we know how the mercury in the floodplain that exists, does it get into the banks? Do we know that mechanism at all?
Well, we've been doing evaluations of the floodplain as part of another project, and the floodplain itself appears to be a very minor contribution, much smaller than current releases from Y12 or the bank soils. And I'm making a distinction here that the floodplain is outside of that bank soil layer. right? So it's primarily relative to leaching from the floodplain or surface erosion from the floodplain, very small percentage related to that. So as far as our evaluation and trying to find solutions for East Fork, we see the floodplain action as being effective at what it did in trying to reduce risk for human exposure. And it makes very little difference relative to how we might approach East Fork. It's that bank and that erosion that we think is the primary driver for the overall flux entering the creek.
Have you folks developed some sort of a formula in terms of you've got mercury in the inorganic form that is ready to, or whatever process nature provides, and how it gets into methylation? Is there some sort of a correlation in there? Like if you got so many tons of mercury available there, there's going to be a percentage of it that gets into to be transformed. Is there any sense of?
Well, the methylation process is a very complicated thing. So there are folks who are within the Office of Science and the basic science programs who are looking very carefully at the methylation process. It's interesting that in places, you have a lot of inorganic mercury, but if you don't have that methylation happening, you don't have risk in fish. Where methylmercury is happening, it seems to be a moving thing. Lately there's a lot of focus on algae, periphyton on rocks. That's a place where methylation is happening. That's a fairly new development. So it's a very, from the science standpoint, what you're getting at is the complexity of the inorganic methylmercury sort of interactions. From a science standpoint, there's a lot we don't know and a lot that's being worked on now. I don't know that it really answers your question.
Sort of does. I guess the other question is I guess, Dave, the design of the mercury treatment facility is sort of ongoing. Mm-hmm. Now, are your folks, your group, part of the process of providing some sort of expertise to this design, or is there no match? Or
Yeah, so the mercury treatment facility, the design is being performed by UCOR, our primary cleanup contractor. And they actually, I think Jimmy Massey's here, one of the gentlemen responsible for that design. And it's standard technology that's being utilized. And it's just to remove the mercury from the water and does not address the methylation aspect. So it's standard technology, nothing innovative being applied, and its purpose is to reduce the amount of mercury flux in the water. Mark, you might want to mention real quickly the challenge we have with the lack of correlation between the concentration of mercury in the water and the methylation in the fish that we've observed over time.
Yeah, that's a really interesting thing. So your higher total mercuries in the creek are up near the facility, and then they decrease with distance downstream. And that's what you'd expect with a point source kind of release, right? You get this mercury coming from the Y12 from various spills, et cetera. And as you get further down, you get more dilution. And so that's the total mercury story, but the methyl mercury is very different. You have relatively low methyl mercury up near the facility, and then that increases with distance downstream. And so if you're trying to clean things up, when you go out and clean up soils or sediments, you don't have methyl mercury in that. It's large. Well, there is some methyl mercury, but if you're doing big soil removal, you're largely grabbing inorganic mercury or elemental mercury. But you can take out a lot of that in the environment, but that may not make a difference relative to methyl mercury.
right, that's interesting.
And so there's just not this nice correlation, that decreasing the amount of cleanup or digging things up more is maybe not going to make a big difference, especially when we get a lot of mercury in the water.
I guess maybe the issue could be solved by us doing the actual physical remediation and you guys coming up with some sort of an answer on the methylation.
Well, you
That
know,
would be great.
Well, I think that's what we're doing. You know, the mercury treatment facility is going to grab a lot of that mercury released from Y12, and then we're going to try to find solutions for the downstream environment. Complex place, but we're hoping to find solutions for there.
Thank you so much.
David Hemmelreich.
Well, thank you, Madam Acting Chair. Mr. Wilson kind of took one of my questions away about the invasive species of mussels that tend to get into streams and are not welcome once they're loose. The methylation that's in the fish, is that generational? Is that passed on to generations through reproduction that you know? Do you know of that?
Wow, you mean, no, I think most methylmercury in fish is totally food chain driven.
Okay.
Yeah.
All right. Thank you.
And there's not a lot of evidence that mercury is toxic to fish. And you've got a lot of very highly contaminated mercury fish in the northern part of the U.S. where you have atmospheric discharge, et cetera. And fish seem to do pretty well themselves. So there's not a reproductive effect.
Okay. Yeah. We had similar conditions in Florida on a hot day. You can go by the canal and just scoop them up.
Yeah.
What is the difference between mercury, which is the liquid metal, inorganic mercury, and methylated mercury?
Well, of course, elemental mercury is mercury zero, and
That's
that's
the
the metal that
Okay.
mercury thermometers,
Right.
beads of mercury. In the environment in East Fork, you don't see a lot of that. It's you do find it around buildings from past spills around the Y12 facility. And then inorganic mercury is mercury salts, mercury sulfide, mercuric chloride, and that inorganic mercury, binds in the environment, and that's where you get cinnabars, mercury sulfide. It's sort of a reddish tint, kind of complex, or kind of rock in the environment. And then methyl mercury is largely generated by microbial organisms, primarily sulfide-reducing bacteria that methylate mercury. And then that methyl mercury, when we're talking total mercury in parts per trillion, methyl mercury is another order of magnitude lower than that typically. So you've got very little methyl mercury in water, but that can drive the food chain, initially starting with algae and then up the food chain, invertebrates, fish, etc. I'm more a biologist than a chemist. I don't know if I got to your...
I'm anything but a scientist, a doctorate, a nuclear physicist. I'm just one of the guys that came in off the street. That's probably the best explanation I've heard. I understand where you're going with it. The last thing I had was a comment. I got some friends in Louisiana. If they knew that crayfish was available, they'd come up and help eradicate it for us. Thank you, Mark.
Keep that in mind.
Alfreda, could I point out one thing? We've got this wonderful map, so I want to use it. Responding to some of the questions that have been asked, if you look at that drawing, the olive green area is roughly the Y12 plant. So that's where all this metallic and inorganic mercury is. Thank you, Dave. The creek, a little higher up, the olive green. Olive green.
There.
Okay, and then the creek flows to the northeast and then roughly follows South Illinois, going up to the northwest now, Dave. Oh, yeah, and then it starts heading, and then you can see the creek, and it flows to the southwest down to ETTP, that brownish area. So when Mark mentions the extent of the fish that have mercury above what we generally consider to be acceptable levels, it continues down to about the ETTP side. The East Fork of Poplar Creek joins the Clinch down there just to the west of the ETTP side. So a big picture, I'm like Mark, I'm a biologist, but I also like pictures. I'm a biologist like pictures. In the big picture, what we have is a metallic and inorganic mercury problem at the Y12 plant, the olive green area, that will be controlled and removed by the D&D program and the water treatment plant. You keep moving downstream, the bacteria go to work, and you get further the downstream reaches where you have more methyl mercury, the organic mercury, that presents the risk pathway because of its absorption in fish. So a lot of the science that Mark and his colleagues are developing might help us solve that problem. But the upstream component is a more straightforward, pull down the mercury buildings, get the mercury out of the water before it leaves the plant. Downstream is where the more I feel more elegant science presents an opportunity to actually reduce the concentrations in the fish. And it's less expensive too, so we've got a two-phase approach going on. Both are important.
Great. Bob?
Yeah, it looks to me like the problem of the source of the mercury in Y12 is soluble, okay? You can stop that ultimately and tear down the buildings, get it out of the sewer system, and get it out of there. But the problem remains then is the downstream part and additional mercury that comes out of the banks of the stream. So as long as it's not going back too far into the banks, though, it looks like you could almost remediate part of the stream by effectively mining the sediment that's accumulated in the banks there and minimize the impact on the stream and do it in piecemeal and solve that over time. That may be wrong, but that's...
I think that's potentially a reasonable thing that may happen. I think
if it's not out in the floodplain...
The evaluation of alternatives, that's a formal CERCLA remedial investigation, and they would evaluate from no action to major soil removal.
Yeah, just look at your current data here. And if it's not out in the floodplain, then your problem is really concentrated right at the stream itself. And that may be a blessing in disguise there ultimately for a solution. But it's long term. To do that take a while.
Okay, since you mentioned methylmercury and it increasing as it flows, as the pathway comes toward ETTP. Okay, we've got residential areas in there. Now, if you're saying methylmercury, then the only time that it would be a hazard to humans is if they eat the
That's
fish or the
Eat a lot of fish.
okay, all right.
Eat
So
a lot of fish.
a lot of fish. Okay, so kids actually playing in the tributaries, that's not a problem. Okay, because we have a whole lot of kids that love that water, and yes, and it's like old time play, and they just have a good time in that water. And I just had to ask that question. Okay.
I think there is, the state maybe can speak to this, there is postings there about no water contact you should be aware of, but it's not related to mercury.
It says mercury on the sign though.
It's not related to mercury. Mercury's not a risk for physical contact in the water.
In the water.
That's right.
Okay. Christoph, do you want to chime in on that, or?
There are postings, and other than mercury, there is some bacterial contamination in a creek. One thing about methylmercury is that it's easily absorbed through the skin. So while we have a small concentrations, that the science is still out on how dangerous that mercury is in a stream. Throughout the years, the concentrations of mercury is dropping, but we still have sources at Y12. We have sources in the banks of the creek. We are in discussions with EPA and with Department of Energy, trying to find the best pathway to remediate that issue.
All right, what I'm taking away from that is that we should caution the children not to play in the water. No, we're not. I'm asking, I'm bringing it up.
The dermal absorption, he's true. Methylmercury would be more readily absorbed through the skin than inorganic mercury. But all the risk assessment work that has been done says that is not a significant exposure pathway. The way you get a lot of mercury in your body is not by sticking your hand in the water, it's by grabbing the fish and eating them, because the fish have concentrated enough that you can actually pick up an appreciable amount. So chemically, yes, it's easier to pass methylmercury through skin than inorganic mercury. In terms of the total magnitude of that transfer, it's still very low, and none of the risk assessment work that's been done says that's an exposure pathway of concern.
Okay, all right, thank you.